Okay, hello everyone. It is Monday evening, October 12th. Hope everyone is doing well. Just some announcements before we get into lecture for today. First of all, today we'll have our lecture on the cell life cycle. And just so you know, I posted a few more Khan Academy videos on Canvas under the lecture module. All of them cover material that we've already covered and will have covered as of the end of this video. I recommend you check those out. They are very valuable. Sal Khan is the one doing those. He goes into some more detail in some areas, less detail in others. You are not responsible for the detail that he goes into that's not covered here in lecture. But especially for some of these more intricate structures and processes that we'll be talking about, that we have been talking about in this unit, I think it's helpful to have a secondary source other than just lecture. Of course, the textbook remains a great source as well. So check out those Khan Academy videos. They are now on Canvas. So this Wednesday, October 14th, we will not have a lecture. So nothing will be posted. But I will still be available for email. If you want to email me to set up a Zoom chat for office hours, let me know. We can meet, and I'm still available. It's an intentional break day that I decided to put in there just to kind of break up the flow of the class, so you're not getting a bunch of material and then having to go straight into an exam. It just felt like it was necessary to introduce a little bit of cushion room into the class in that way. So this Wednesday, the 14th, no lecture. Please use the time for studying and so forth. Lab also, along that same line, lab this week is going to involve you visiting a natural field site of some kind, and I'll include some ideas. We've already talked about some ideas already. I'm still working on the instructions document, but just because it does involve you making a trip, and so for some of you that the weekend might be the best time to do that. Also, we have an exam coming up next week. I decided, and really should have done this from the beginning, to make this another 2-week assignment. So you won't have lab due next Monday. Instead, the Natural Field Reserve journal entry assignment is going to be due in 2 weeks, which is Monday, October 26th. So near the end of October. So again, nothing will be due next week. And that will give you more time. It'll be worth a regular lab, unlike the urban ecology lab, still worth 20 points. No, let me figure that out. I may take that back. But again, just like with the urban ecology lab, which coincided, if you'll remember, during exam 1, I don't want you to have a huge assignment due, during an exam week. I just don't think that's fair. So that's why we're doing it that way. I sent an email out a little bit earlier on October 19th, which is next Monday, a week from this evening. We're going to have a study session, same format as last time. And thanks for those of you who filled out the poll online indicating your dates. As is laid out in the email, there was an extremely even distribution of votes. I didn't expect that. I kind of expected it to be leaning more one way or the other. And so I issued a tiebreaker, and we're going to go ahead with the time that we did it last time, which is the normal time that we do a lecture, a live lecture, 8:30 p.m. on October 19th, Monday, October 19th. So I'm sorry if that's a time that you're unavailable. I understand. It's just a limitation of the online environment. It's just an opportunity for you to ask questions that maybe you're not getting adequately during the recordings. I know that this medium is kind of limiting in that way. And even when we were having online, if this were a physical class, we'd be doing the same thing. I just think these study sessions are helpful. So I'm sorry if you can't make it, but let me know if you want to have a Zoom session to go over any questions that are specific. I'm still willing to meet with you one-on-one if you can't make it to the study session. The study guide actually is already posted on Canvas for exam 2, so take a look at that. So the Wednesday after October 19th, so that's the 21st of October, that will be the exam 2 date. So that's a week from Wednesday, about a week and a half from now. So let that be on your horizon. Remember that there's a pretty narrow time window that the exam is workable, from 5:30 to 8:30 p.m. on Canvas. So please plan accordingly to be there. I would say that the exam would be about an hour in length, but you might want to give yourself some cushion room. I recommend starting at 5:30. Okay, with that said, come on now. Not listening to me. Oh, there we go. Okay. So just a recap on what we talked about last time. We talked about cell structure, cell function last time, but I think it's helpful to kind of go over cell structure and function, since they are really two sides of the same coin. So question one is, osmosis is a type of what? And we actually didn't talk about all these processes this semester. A little bit too many moving pieces. But we did say that osmosis is simply the movement of water across the cell membrane, in which case it's a type of diffusion. So as water moves across the cell membrane down its water potential through that process we talked about last time, that is a process of diffusion. Anytime water moves through something, it's diffusing through that something. So when water goes through the cell membrane, whether it's going from the outside of the cell in or the inside of the cell out, that is diffusion. Question 2, which way will the water move? Well, we posited last time that the potential energy for water, which is this term water potential, always goes from states of high potential energy to low water potential energy. And there's a bunch of different things that affect water potential, but in a really broad way, where there is more water, that's where the water potential is higher. And water potential always goes from high water potential to low water potential. So if you look at this schematic here, and you imagine these little blue balls are water molecules. Water, of course, is a solvent of life. And these red balls are some kind of solute. Let's call it salt, just to keep it simple. And this black dashed line is the cell membrane. Okay, so the outside of the cell, perhaps, and the inside of the cell. Okay, water potential always goes from a state of high water potential to low water potential. And the water potential is higher where there's more water. Where is there more water? On the right side, or in this case, the inside of the cell. And where there is more water, the water potential is higher. So where does it go? Always from high to low. The water will move that way. So that's the actual chemical, physical characteristic that determines the direction of osmosis, is water potential. Which one of these is not a domain of life? This is, we talked about the domains of life in the context of the Linnaean system of classification. One of these should stick out to you as definitely not belonging. Everything we've been talking about so far pertains to one of these three domains, but this one, I just made this term up, oologia. That doesn't really, it's completely bogus. So the three domains of life are, of course, the eukarya, the archaea, and the bacteria. And we, of course, have been spending the vast majority of our time in the eukarya domain. Which structure do plant cells have that is lacking in animal cells? Or which structures, so the plural, okay? Well, let's just go through these one by one. Ribosomes, of course, are the factories of cells, and that's where cells manufacture their proteins. And animal cells and plant cells both need proteins, so they both have ribosomes. Whereas cell membrane is another term for that is the plasma membrane or the phospholipid bilayer. That's the organelle that divides the inside of the cell from the outside of the cell. It's what we talked about in the context of water transport and osmosis. So, you know, a lot of last lecture, we talked about that in terms of cell function. A very important molecule or membrane that, you know, the molecules that make it up are, of course, phospholipids. Both plants and animals do have cell membranes. Okay, they both have phospholipid bilayers. But, you know, and this is where it's kind of confusing because these two terms sound the same, although they are very different. Plants have an additional structure that's actually on top of their cell walls or cell membranes called the cell wall. And chemically speaking, it's very different from the cell membrane. It's made of carbohydrates, a specific type of carbohydrate polysaccharide called cellulose. That's what helps give plant cells their rigidity. And so if you look at plant cells under a microscope, they're kind of box-shaped, they're rigid, and they're very interlocked. In large part, that has to do with the cell walls that give plants their structure. And chloroplasts, of course, are organelles where it's the site of photosynthesis, and those are more or less endemic to plants. They only occur in plants with a couple of weird exceptions, like some algae that are pseudoplants, but they certainly do not occur in animal cells. Okay, so cell walls and chloroplasts only occur in plant cells. And last question, prokaryotes are blank in comparison to eukaryotes. Okay, so we've talked about these two cell types, prokaryotic cells, which are in the domains bacteria and archaea, that we acknowledged at the beginning of this unit, but we didn't go into much detail into. And then eukaryotes, which are basically everything we've been talking about in terms of cell structure. Okay. Broadly speaking, in terms of comparing these two types of cells to another, prokaryotic cells are generally smaller and they have less internal organization relative to eukaryotic cells. So generally speaking, they're several orders of magnitude smaller than eukaryotic cells. You know, things like bacteria, E. coli, for example. And they don't have a lot of these internal organelles that we've spent so much time talking about. The nucleus, the mitochondria, the endomembrane system, okay? They don't have a lot of these internal organelles. They do have things like ribosomes, they do have DNA, but they are more or less kind of dilute in the center chamber of the prokaryotic cell, whereas there's a lot more neighborhood organization inside of eukaryotic cells. Eukaryotic cells are much more like cities in that sense. And we used cities as a kind of a euphemism for how eukaryotic cells are put together. Okay, with that, we're going to go ahead and dive into the cell life cycle, which is found in chapter 8 of your textbook. And I'll say again that Sal Khan also does an excellent video, which is now on Canvas, that I recommend you check out in addition to this lecture, because there are some, what I'm going to talk about, it's not complicated, it's not hard, but it's just kind of detailed. And so, I'll give to you some helpful tips that I think can help you get your brain around some of the subject matter. It's not complicated. If I can do it, you can do it, but it is kind of persnickety, I'll say. And I'm sure you've seen some of this before in high school biology. So let's just take it really slow. I'm sorry that we can't be in person because it can be a lot easier to learn this material if we were all together. But let's just take it really slow. Feel free to rewind this as many times as you like, and watch Sal Khan's video as a compliment because it is a good compliment to what I'll talk about. Okay, and just getting back to the properties of life. This is the same slide that you've seen on several occasions now, and we've been exploring various aspects of these properties of life throughout the whole course. We've examined various aspects of order and regulation, a little bit on growth. We have examined a little bit on energy processing. We'll do even more of that later. We've definitely examined responses to the environment and evolution. We've examined reproduction. I mean, we've examined these properties at one tier or another already in this course, but we're going to continue to revisit them at various stages. Certainly today we're definitely hitting on reproduction, but we're also talking about growth and development. We're also talking about energy processing. We're even talking about regulation to a certain extent. So this is just a reminder that life is, insofar as these properties are concerned, they're all interrelated. But we're talking about the cell life cycle. Of course, we're talking about how cells reproduce themselves, which is obviously a very biologically important thing. I mean, we wouldn't be able to live if our cells did not have the capacity to reproduce. So it's pretty important. So again, this is something that looks kind of wonky the first time you see it, but let's just take it really slow, and you'll see that this is not that. It's pretty straightforward. Okay, so this whole thing here, this whole schematic, this is the cell life cycle. Okay, and it's circular. Gosh, my words are all messed up tonight. When something it's cyclical. There we go. That's the word I was looking for. It's cyclical, meaning that it's seasonal. It goes around and around and around. That's the cell life cycle. Okay, and biologists have broken down the cell life cycle, the cyclical life cycle, into various phases. Okay, and that's what's denoted here by these black letters. Okay, I'm not going to ask you to memorize these letters, most of them in any intricate detail. Again, this is just something that biologists use to try to very broadly get a handle on what's going on. They're not necessarily set in stone as extremely distinct periods within a cell's life. Okay, but there are two phases of the cell cycle, broad phases that I do want you to be familiar with. The first phase is called interphase. Okay, so interphase is the period of the cell when it's not actively going through cell division, when its nucleus is not dividing. Okay, so that's shown here by the orange, the green, and the turquoise colors here. So the G0, G1, S, and G2, these sub-phases, if you will, which I don't expect you to memorize. I do want you to know that this is interphase, which is simply the phase of the cell when it's just going about its regular business. It's not actively dividing. So this is a period of the cell's life when it's growing, it's going about its regular business, its organelles are up to their usual defined processes. The DNA is translating itself into proteins, and it's regular life. It's the 9 to 5 for a cell. But when a cell gets to a point where it's ready to divide, it's an elderly cell, and it's reached the end of its particular life cycle, and it's ready to reproduce, to have offspring. That is this M phase of the cell. Okay, so all of this, 2/3, most of the cell's life cycle in terms of time spent is interphase. Okay, whoops. Most of the cell's life is interphase, shown right here. But eventually, the cell gets to a point where it's ready to divide. That's shown here by this red slice of the pie. And this is the M phase. Okay, so this is when the nucleus is actually dividing, and cell division is occurring. Okay, so the whole thing is the cell life cycle, but we've delineated so far between interphase, which is the regular 9 to 5 of cells, and the M phase, which is the cell division, the nuclear division. phase of a cell's life. Okay, so when we're talking about cell division, which is only one part of the cell life cycle, we're honing in specifically on this red slice of the pie. And there's different subphases within the M phase, which I do want you to know. But again, it's pretty straightforward. If we take it, remember, this is cyclical. You always know what's going to happen next. Okay, so cell division. Here's an electron, actually a compound light microscope image of a cell that's going through division. It's a pretty neat fluorescent picture. Okay, so this is a cell that's certainly in the M phase. It's actively dividing. It's actually a specific subphase of cells, which we'll talk of cell division, that we'll talk about here shortly. But this is what you might expect to see it look like. And just to be clear, we are still confining ourselves to eukaryotic cells. We're not considering prokaryotic cell division. Prokaryotes certainly do divide themselves, and they do have a cell life cycle. And there are various similarities, but there are also some differences. And so I just don't want to, it's just too much to consider both. We're constraining ourselves to only eukaryotic cell division as it is currently understood. Okay, so everything we're talking about only occurs in eukaryotes. And there's a couple types of cell division, the M phase, if you will, of the cell life cycle. Okay, the first type is asexual, meaning when there is no exchange of genetic material, where it's just basic clonal reproduction. And a lot of organisms do this. Their cells reproduce asexually, and they basically clone themselves. We'll run through some examples of those. Okay, so when cells asexually divide, that is called mitosis. So M-I-T-O-S-I-S, mitosis. That is asexual reproduction, where basically two cells, which are clones, come out of one parent cell, and they're genetically identical to the parent cell. Okay, that is mitosis. It's asexual cell division. The other type of cell division is sexual, as you can imagine. And so this is where there is actually exchange of genetic information between, two parents, and there are new combinations of genetic code, and we'll talk about that process actually in the next unit. So when cells divide, in order to get ready for sexual reproduction, this process of cells getting ready for that is sexual cell division. That is called meiosis. So meiosis is sexual, where 4 cells are produced that are distinctly designed for genetic exchange. Okay, so just as a recap, we're considering 2 types of cell division. The first type is asexual, called mitosis, where one parent cell reproduces 2 what are called daughter cells. I don't know why daughter was chosen as the biologists name things. Could have easily just have called it son, but they're 2 daughter cells. That's how it's referred to, and they are genetically identical to each other and to the parent. Asexual. Whereas sexual cell division is the process called meiosis. So I know these 2 terms sound very similar, but mitosis and meiosis. Mitosis is asexual, meiosis is sexual. And in the context of meiosis, this is a cell getting ready for sexual reproduction. It's producing cells like sperm and egg cells that are specifically designed for sexual reproduction, where not just 2 cells are produced, but actually 4 cells are produced that only have half the amount of DNA relative to the original cell. And we'll talk about why that is in a little bit. So we're firstly going to consider only asexual reproduction. Okay, so mitosis. Okay, this is the process by which cells clone themselves. Okay, and if we extrapolate up to the organismal level, we're considering this for cells, and we'll talk about the cell process of asexual reproduction here shortly. But if we just take a liberty here for a second and extrapolate up to the organismal level, organisms, whoops, so I'm getting a little bit ahead of myself. I don't have my preview in front of me. Anytime, so keep that in mind what I just said. We'll come back to it very quickly. Mitosis, anytime a parent cell splices itself in two and creates two genetically identical individuals, that is asexual, of course. And the cells that do that, they're not splitting their DNA into, the genome from one cell to the other cell is exactly the same. There's no division of the DNA. So these cells that have a full complement of DNA, they're called somatic cells. Okay, so mitosis produces somatic cells, which are simply cells that are clones of the original parent. They're not sex cells. Okay. Okay, so getting back to what I said, if we extrapolate up the process of mitosis to the organismal level, if we kind of walk away from cells here for a second, organisms reproduce asexually all the time. So even single cellular eukaryotes like little amoebas, this is one cell large, they of course reproduce asexually, so it's dividing here. But then multicellular eukaryotes reproduce asexually all the time as well. Sea stars, for example, are very well-known asexual reproducers. If you take a sea star, it sounds cruel, don't ever do this, but if you take a sea star and cut it in half, odds are you've helped it reproduce. You haven't killed it. Those two halves of the sea star will very often butt off and become separate individuals in and of themselves, even though they came from the same parent. So that's the organismal manifestation of mitosis. Violets are another example. African violets. If you take the leaf and the petiole off of, not just violets do this, lots of plants do. Take the leaf and petiole off an African violet and plant it back in the soil. That cut section will often shoot up roots, and it will become its own plant, even though it's basically the same as the parent. So other things that are very commonly reproduced through asexual means, and this is kind of at the crop level. So a lot of bananas, that we very commonly eat as part of our diet. If you consider places where bananas are cultivated, which really is not the United States for the most part, but down in Central and South America, where this is a very staple crop, a lot of bananas are grown. Banana trees are grown from cuttings, where banana farmers will cut portions off of the banana tree, and they'll plant those cuttings in the ground, and that will form a new banana tree. Clearly, there was no exchange of pollen, so that new banana tree is genetically identical to the parent. And a lot of crops are actually reproduced this way. Strawberries and potatoes, a lot of very common crops. Pineapples, peaches, avocados, grapes, blackberries, a lot of things that you'd see in your garden. They reproduce asexually. They don't, they can still reproduce sexually, but cultivation-wise, they are often reproduced asexually. If we get to a more natural process, we've talked about this before a little bit already, I think, but these quaking aspens, which I think these are from Switzerland. This is obviously a helicopter view, but all of these trees right here that it looks like a forest, right? Well, it's all one individual. It kind of looks like this underneath the surface. If you were to look underneath the soil, there's like one parent tree that has roots growing parallel to the soil underneath. And from that parallel root growth, a new shoot might emerge and become, kind of a little subordinate parent tree, and then other little subordinate child tree, and then other little subordinate trees will keep growing and growing. Eventually, these roots will butt off, and this individual and this individual and this individual, they'll all butt off from one another, and they will be functionally their own individuals, and yet clearly the result of asexual reproduction. That's what we're seeing here. This is all from one individual that shared the same root space in the soil. So that's, ecologically, it's a pretty common phenomenon, especially for plants to go through this. Joshua trees are another example, at least the subspecies we have here in our county, in Kern County. The subspecies of Joshuas very commonly reproduce asexually, where they, the same process is right here. They send sections of roots lateral across the surface, and then they bud off from that. They still have the ability to produce flowers and fruits and so forth. They still have the ability to reproduce asexually, but often they choose to reproduce, or they have the capacity to produce sexually, but often they choose to produce asexually instead. And we'll actually talk in the next unit about why plants sometimes choose one path over another. Okay, these are just, it's kind of preaching to the choir a little at this point, but strawberries, they'll send these parallel branches across the soil of the surface. These are called stolons that can bud off and to create new individuals. So look right here. These are the same individual, but if you were to cut off this branch right here, these two things have roots and they can become their own individuals. So a lot of other things like ginger, for example, is another root that can reproduce and bud off asexually. So anyway, so that's kind of an organismal level of how cells can reproduce themselves. So if we go back to the cell level, okay, we looked at the organismal level. Let's microscope back into the cells. We're considering how eukaryotic cells asexually reproduce themselves through the process of mitosis. Okay, so if we're considering how cells do this, we need to kind of even hone in a little bit further than a whole eukaryotic cell. We need to hone in specifically on the nucleus. Okay, the nucleus, which is the manager of the cell. If you remember from the plant or from the cell structure lecture, the nucleus is in the center of the cell, and it's where the DNA resides. It's the corporate headquarters, if you will. Okay, and we'll talk a lot about DNA in the next unit, so we're not going into too much detail here, but we do have to have a reasonable understanding of the structure of DNA in the nucleus of a cell in order to understand the process of mitosis. Okay, so here in this artist's rendition, I know there's a fair amount going on right here, but this is a nucleus right here. We're not looking at the cell, we're zoomed in even further than that. This is the nucleus, okay? And so here's the nuclear membrane, okay? And all this spaghetti-like stuff in the center of the nucleus, that's the DNA, okay? And inside the nucleus, the DNA, it can take on a variety of different forms, okay? It all just depends on the way it's packaged. No matter what, it's still DNA, okay? It's all still the same thing, but it just can repackage itself into different forms depending on the stage of the cell life cycle. So when a cell is going through interphase, the regular 9 to 5 of the cell life cycle, and it's just going about its business, it's not actively dividing, the DNA inside the nucleus exists in this dilute state. Okay, it looks like spaghetti, in this particular artist's rendition. It doesn't really look like spaghetti if you were to look at it under a microscope, but this is an artist's attempt to try to get a handle on it. So when the DNA in a nucleus is dilute inside during interphase, this spaghetti-like structure, that is called chromatin. Okay, so that's the form, the packaging that the DNA is in during interphase, the regular majority part of a cell's life. However, when a cell starts to move into M phase, whether it's mitosis or meiosis, but we're talking about mitosis right now, when a cell gets ready to divide its nucleus, It's just kind of like whenever you have to move. What do you have to do when you have to move? You have to pack up all your stuff into boxes. So DNA, whenever it's getting ready to move, whenever the nucleus is getting ready to divide, the DNA has to pack itself up. Okay, so the DNA, it's a really long molecule. It's one of your cells has enough length to go to the moon in terms of one DNA molecule. These are really, really long molecules. But it has to, when it exists in this chromatin-like state when it's going through interphase, but when it moves into M phase, it has to package itself up for the move. Okay, so it winds itself around these proteins called histones. This is a little bit TMI, but ultimately it packages itself very finely into a structure that you've probably heard of before called a chromosome. Okay, a chromosome is simply the condensed form of DNA. So just as a rehash, when a cell is in interphase and going through its regular kind of 9 to 5 duties, the DNA exists in a dilute state in the nucleus. That's called chromatin. When a cell is getting ready to divide itself, it has to package itself up. And as it packages itself up, it forms this structure called a chromosome, which is simply the packaged form of DNA. So packaged is chromosome, unpackaged is chromatin. I know these terms are a little bit wonky, but that's just what they're called. I'm sorry, I wish they called it unpackaged DNA and packaged DNA, but this is just the way it is. This will make a little more sense as we go on. So here's a chromosome, which again is the packaged form of a DNA molecule. Okay, and this is again an artist's rendition, of what a chromosome looks like. And if it weren't complicated enough, in this artist's rendition, if you consider the whole thing, all this broad X shape, the whole thing is called a chromosome. But each half, if you were to split it in half, each half is called a sister chromatid. Okay, so I know there's a lot of chromos going on here. We have the chromatin, okay, the unpackaged DNA. Then we have the chromosome, which is the packaged DNA. And then we have the sister chromatids, which are each respective half of a chromosome. I know that's wonky, so just watch this video a bunch of times until it makes sense. I know it's not complicated, but it's weird. So I'm sorry that there's just no other way to get a handle on it, but that's just the way it is. Again, so chromatin, chromosomes, and chromatids. There's these three very different sounding terms. Well, they're similar sounding terms that are different things. Okay, so this is a chromosome. So here is an actual electron picture. Well, no, not an electron. This is a light microscope picture of an actual plant cell. Okay, so here is an individual plant cell. This blue structure here is the cell wall. You can see the cell wall really nicely, can't you? And here's the nucleus, okay, in each plant cell, okay? When a plant cell, or any cell for that matter, is getting ready to divide, going into the M phase, it forms chromosomes. So can you see the distinct chromosomes forming here in this plant cell? This is a cell that is getting ready to divide itself. But if you look at these neighboring plant cells, the nuclei in these respective neighboring plant cells, the DNA is dilute inside the center of the nucleus. And so that is chromatin. Okay, this is the same thing. It's all DNA. It's just the way that it's packaged. Okay, with all that said, we're going to dive now into the phases of mitosis, the M phase, okay? And this is where people can get a little bit wonked out because there's, some wonky terms. But just keep in mind, it's a linear process. It's a circular process. It always repeats itself. And once you kind of get the flow of it, it's actually pretty straightforward. Okay. So we're going to talk about these phases of mitosis, which I would like you to be able to know and to be able to describe in your own words. The first is called, well, first of all, I'd like to just give you a little acronym to remember here. Just like with the organic compounds, the organic molecules rather, the organic elements. Remember we talked about SHNOPS, carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. You can remember the organic elements very easily that way. There's a similar little thing that you can use to remember the phases of mitosis. That's PMAT-C. If you can remember PMAT-C, you can describe the phases of mitosis with relative ease. Okay, so refer back to this slide for a full kind of written description of these various phases, but it makes much more sense if you can visualize it in a picture. So we're going to explore these phases, and remember, this is a story. It's a season. It always just keeps going around and around. Okay, so again, these are the phases of mitosis, and ignore this one right here, because this is actually an extra step that we're not going to bother with. So PMAT-C. Okay, so P is the first letter that we're concerning ourselves in the phases of mitosis. So P is prophase. Okay, and this is the most complicated phase. The very first one is the most complicated phase of mitosis. So if you can get this, everything else is a breeze. There's 3 things that go on during prophase. Okay, it's the first thing that happens. So a cell, it's going through its interphase, which is the vast majority of the cell cycle, and the cell decides, okay, I'm ready to divide. Okay, I'm old, I need to reproduce myself. So I'm going to go into M phase now, the mitosis phase. The very first phase of M phase is prophase. Okay, so PMAT-C. And there's 3 things that happen during prophase. First of all, the chromatin condenses into chromosomes. Okay, remember chromatin is the uncondensed, the dilute form of DNA during interphase. So the first thing that happens during prophase is the chromatin condenses to form chromosomes in the nucleus. So the DNA packages itself. Okay, it condenses itself. That's step 1 of prophase. Step 2 is that the nuclear membrane breaks down, okay? Or at least it begins to break down. Okay, so the nuclear membrane, that's what we're visualizing right here. We're not looking at cells, we're looking at a nucleus. Okay, the membrane that surrounds the nucleus begins to disintegrate. Doesn't completely disintegrate, but it begins to disintegrate during prophase. That's the second thing that happens. And thirdly, this is kind of more weird terms, but they're not, if you visualize it, it's not that complicated. These long fibers attach themselves to the chromosomes. Okay, these fibers are called spindle microtubules. Okay, so this actually is part of the same organelle structure. Remember we talked about the cytoskeleton, which is one of the organelles we talked about for cell structure. These spindle microtubules are part of the cytoskeleton. The cytoskeleton is the support network within the cell. It's the internal framework, if you will. Spindle microtubules are part of the cytoskeleton. Okay, so the third thing that happens at the chromosomes, the spindle microtubules, which are these fibers here, attach themselves at certain parts of the chromosomes. Okay, these certain parts are called the kinetochores. Okay, and again, I apologize for, I don't like to throw a lot of vernacular at you, but it's the way it is. It's, there's no other way to define it. There's no other term. Okay, so let's just go through that again one more time, because I know people people struggle with this. I struggled with this when I first learned it. So let's just go through this again. During prophase, which is the very first phase of mitosis, there's three things that happen. The first thing that happens is that the chromatin condenses into chromosomes. Okay, so the spaghetti-like structure that was there during interphase, as a cell is entering M phase, the chromatin condenses into chromosomes. Okay. Secondly, the envelope, the membrane that surrounds the nucleus, begins to break down. And thirdly, the spindle microtubules attach themselves at the kinetochores, the central portion of the chromosomes. Okay, so that's all shown right here. Chromosomes condense, nuclear envelope breaks down, and these long fibers attach themselves to the chromosomes. Okay, so that's kind of shown here by this specific image. Okay, if you're with me so far, you've got this now, because that was the most complicated part of mitosis. The next few phases, very simple, relatively speaking. So P, the first phase of mitosis, is prophase. We've done that. Check. P, mat, C. What's the next letter? M, metaphase. Okay. So during metaphase, only one thing happens. These spindle microtubules have, remember, attached themselves to the kinetochores of the chromosomes. Okay. So these spindle microtubules, think of them as long ropes that are kind of pulling ships into a port. Okay. These spindle microtubules are attached at respective ends of the cells, and they kind of put the chromosomes under tension. They begin to pull. And during metaphase, the chromosomes line themselves up at the center of the cell. Okay. Some people call this central thing here the metaphase plate, although there really isn't any kind of firm structure involved. It's just a period in time. All of these phases are just periods in time. They're not hard and distinct. Okay. But metaphase is simply that period in time when the chromosomes are kind of just lined up at the center of the cell. They haven't split yet. That happens next. So P is prophase. We've done that. M, metaphase. That's when the chromosomes have lined up at the center of the cell. PMA, anaphase. Anaphase is when now the spindle microtubules are pulling pretty hard on the chromosomes, and the chromosomes actually split. Okay, the chromosomes split and begin to move towards opposite ends of the cell. Okay, and this is kind of a weird thing. Okay, let's go back to this right here. This whole thing is called a chromosome, if you'll remember. And when a chromosome is in one big piece like this, each side is called a sister chromatid, right? So sister chromatids, and the whole thing is a chromosome. But during anaphase, when the spindle microtubules pull the chromosome apart, the results of each half are no longer called chromatids at this point. Functionally speaking, now we're going to call each one of these two halves their own chromosomes. Okay, so each one of these, what was previously a sister chromatid, now they're called daughter chromosomes. Okay, it's just the way it is. So during anaphase, the chromosomes split apart, and the daughter chromosomes are now moving toward the respective ends of the cells, of the cell. So PMA. And then TC. I'm kind of combining them together here because it makes a little more sense. So during anaphase, the chromosomes, as you can imagine, are kind of moving towards the respective ends of the cells. During telophase, the chromosomes actually reach their destination, which is the respective end of each cell, and the nuclear envelope, the nucleus, reforms around the two halves of the of the two parts of the nucleus, okay, or the two parts, the two ends of the cell. Okay, so the nuclear envelope kind of reforms, and this last phase, which kind of doesn't have a phase attached at the end of it, is when it's called cytokinesis. This is when the cell membrane actually begins to start to butt itself off. Okay, that's shown here. Here's another picture, an electron microscope of the cell membrane, not just the nucleus, but the whole cell membrane beginning to form this little furrow here. This is the process of cytokinesis. Ultimately, after cytokinesis, two daughter cells, which are genetically identical to the parent cell, butt off. Their chromosomes have arrived at opposite ends of the cell. The nucleus has reformed. All the organelles have now reformed. And these are two individual cells that are genetically identical to one another. Okay, so just to go, I know that's a lot. So just to to rehash it one more time: PMAT-C, prophase, metaphase, anaphase, telophase, and cytokinesis. During prophase, the most complicated phase, the chromosomes condense out of chromatin, the nuclear envelope breaks down, and the spindle microtubules attach themselves to the kinetochores. Try saying that 10 times fast. But once you visualize it, you know what's happening. You've seen the images. Okay, so that's prophase. Next, metaphase. The spindle microtubules put tension on the chromosomes, and they are kind of lined up at the center of the cell. During anaphase, the chromosomes are split in two, and the daughter chromosomes are now moving toward the opposite ends of the cell. During telophase, the daughter chromosomes have arrived at the opposite ends of the cell, and the nuclear envelope, the nuclear membrane, reforms around each of those sets of daughter chromosomes. And during cytokinesis, the cell membrane actually buds off and splits into two new cells. That's shown here, okay? That is the process of mitosis. I know it's kind of a mouthful, a lot of vernacular and jargon, but if you can visualize it, don't think so much on the terms, but rather visualize it. It's very linear. It's easier to understand if you just watch it. So so there he is. So again, these phases that we've been talking about, they're just snapshots in time. They're not like the cell's not thinking to itself, okay, now I'm going through anaphase, or now I'm going to go to telophase. The cell doesn't think of it that way. The cell is they're just periods in time that we biologists have, classified these various phases so that we can try to get an understanding ourselves. Okay, these are just categories, if you will, that we've used. Okay, so getting back to the organismal level, mitosis is always occurring. Okay, it's how all the cells in your body, with the exception of your sex cells, your eggs or your sperm, whether you're male, female, so the cells in your skin, the cells in your blood, the cells in your kidney, all the cells that are not involved in sexual reproduction, they're always going through the process of mitosis because our cells get old and they have to divide themselves. That's how complex multicellular organisms such as us maintain our cellular integrity. So mitosis is always occurring. So here, for example, is a cross section of an onion root, and we would actually do this if we were in lab. We had microscopes. We would actually make our own microscope slides of this. But these are, each one of these little boxes is an individual plant cell in an onion root, and you can see all these different phases of mitosis all going on at the same time. Some cells are in prophase, some cells are in metaphase, some cells are in anaphase. You can see very distinctly some cells are in telophase, and you can even see a little bit of cytokinesis going on here. Mitosis is always occurring. It's just a natural part of life. It's all part of the cell cycle. Okay, Now, we've gone through sexual or asexual reproduction in the context of mitosis. We're really quickly going to hit on the process of meiosis, which is sexual reproduction. Okay. Meiosis, you know, as we talked about, it's the process of actually producing cells that are capable of sexual reproduction. So it's not the sexual act itself, and we're not going to go into sex ed here. This is not the purpose. But meiosis is the actual process of creating sex cells. Okay, so these are called the gametes or germ cells. Okay, so you know, unlike somatic cells, which is the product of mitosis, where clonal individuals, two daughter cells, are formed out of one parent cell, during meiosis, sex cells are produced that have half the amount of DNA from the parent cell. Okay, so they are not somatic cells, they're germ cells. Okay, so biologists often differentiate here by having a half complement of DNA versus a full complement of DNA. And we'll talk about this a lot in the next unit, but little n here versus 2n. Okay, another way of saying this is haploid, ha kind of meaning half, and diploid, di meaning two. Okay, so somatic cells are diploid, they have a full complement of DNA. Germ cells are haploid, they have a half complement of DNA. Okay, and so we're not going to go through all the phases of meiosis like we did with mitosis. I just think that's unwieldy. You're already learning enough. But the easiest way to think of meiosis in a very broad sense is that it's kind of like mitosis times 2. Okay, so there's 2 phases of meiosis. Meiosis 1 is basically mitosis. Okay, I'll say that again. Meiosis 1 is basically mitosis, where a parent cell divides itself and produces 2 genetically identical, daughter cells. But then, during meiosis 2, those daughter cells then split themselves again. Okay, and their DNA actually splits itself in half. And so you end up with 4 cells that are distinct from one another, genetically speaking. Okay, because their genomes have essentially been ripped in half. Okay, so in a broad sense, meiosis is like mitosis times 2, but we're not going to go into the prophase 1, metaphase 1, anaphase 1, all that. It's just TMI. A very important aspect of this, though, is how sexual reproduction occurs. As one of the phases of meiosis, prophase 1, there's this very, very important, this is really the whole point. The whole point of meiosis is sexual reproduction, new combinations of genetic information. So during prophase 1, I would like you to know this, there's a very important process that goes on called recombination, which is where, 2 chromosomes, not within a chromosome, but 2 actual separate chromosomes actually cross over. This event is also called crossing over, and actually exchange genetic information, and ultimately end up having brand new, completely novel sequences of DNA. That's the actual sexual reproduction there. That's why you and I, which are the offspring of sexual reproduction, we're not clones of our parents, right? We are our own genetic individuals. That process is thanks in part to recombination during meiosis 1, where respective chromosomes actually cross over and create completely novel sequences of DNA. That's what makes us, at the genetic level at least, distinct individuals. Okay, so that's a complete mouthful. I know that's a lot we've been talking about so far. We talked a lot about mitosis, which is asexual reproduction, and we've talked very briefly about meiosis. Here is a helpful chart, but again, just as a very quick rehash, mitosis is asexual. It produces 2 genetically identical daughter cells, whereas meiosis is sexual. And through the process of 2 phases of meiosis and recombination of chromosomes, you end up with 4 genetically distinct germ cells that are then ready for sexual reproduction. Okay, with that, that is enough for this evening. So I'm going to go ahead and sign off for now. But watch that video as many times as you like. Remember PMAT-C. I know it's a lot of nomenclature, so I apologize for that, but that's the most nomenclature I will ever throw at you in one lecture. So watch that multiple times. Also watch Khan's video on the cell cycle. That will help as well with trying to wrap your head around these concepts and processes. With that said, keep in mind the announcements for today, and I will see you all, well, actually, I will not see you on Wednesday because we have a break day. So keep an eye on Canvas for the field trip to the Natural Reserve Site lab instructions, which I'll probably post sometime tomorrow, which is Tuesday now. And other than that, let me know if you want to meet on Zoom or just send me an email message. Because we're not going to meet on Wednesday for lecture, but we will have a study session a week from today, which is the 19th at 8:30 p.m. So I'll be in contact between now and then. I'll send some emails just to remind you and everything. But please, on Wednesday, don't hesitate. You know, I am still 100% available to you. If you have any questions or concerns, my door is open, my virtual door, that is. So please let me know if you need any help. I would love to meet with you on Zoom to go over anything that you like. With that said, I'll sign off for this evening. Have a good one, everyone.